I'm switching on the recording of the meeting, and I also switching to English. First and foremost, we have a number of new people who are joining us. So lectures 2 and 3, both handouts and the records of the lecture are in a Dropbox. I hope that Natalia Viktorovna Remke would help to get it. We will also place there the handouts from the lecture 1 about imaging modalities, and also hopefully Anna has recorded it, so then there will be full text. The topic of today's lecture will be radiometal-based radiopharmaceuticals, but I would like to make a reservation. Probably the hottest one, PSMA binder, would not be touched during this lecture. So Professor Orlova agreed to give a separate lecture concerning prostate cancer imaging, and this topic deserves indeed the whole story. But we gonna talk about radiometals today. My question is, do you see my pointer? Somehow.
Yes.
All right, excellent. So because I need it. All right. During our previous discussion, we pointed that radiometal or metals are the most common elements, and accordingly, the chances to find the radionuclide with suitable chemical and physical properties are, of course, the highest among the radiometals. And there are a variety of nuclides which are potentially suitable. However, it's only a few nuclides found the application in day-by-day clinical use. That includes two radionuclides, gallium and indium for imaging. It's yttrium, high energy beta emitter, which is used for therapy, samarium, which is used for bone pain palliation, and lutetium, low energy beta emitter, which is used in the therapy. There are other nuclides which are used in clinics pretty actively. For example, rising star is actinium or radium, which are alpha emitters, but it's still rather exploratory work for actinium, and for radium it's not a true targeting and no chemistry involved. It's just a chloride. And technetium will be considered separately as well. I would like to start with gallium, which is one of the most deserved radionuclides in radiopharmacy. And historically, the first nuclide which has been used in oncology imaging is a gallium-67. Before FDG era, it was a workhorse of nuclear oncology. Not really ideal nuclide. 300 keV gamma is a little bit on the upper borderline. 93 keV is a bit too low, and a lot of radiation would be scattered, but still it is acceptable for imaging. There is also nowadays rising star, gallium-68, which is a positron emitter with a half-life of 67 minutes, sufficient for peptides, sufficient for scaffold protein half-life imaging. The major advantage is that it is a generator-produced nuclide. So it's a daughter of germanium-68, and mother nuclide is immobilized on the ion exchange column, and every 4 hours you put milk away, elute daughter nuclides with activity approximately equal to mother nuclides. So more and more gallium-based radiopharmaceuticals entering the market. Most important was that approximately 3 years ago, European agency, medical agency approved the gallium generator for production of radiopharmaceutical. One more interesting nuclide is gallium-66. It is also positron emitter, which means that one could use it for positron emission tomography when you work with a bit more long-lived radiopharmaceuticals which require a bit more time. So time window for gallium-68 is approximately 3 half-life. It's between 3 and 4 hours. If, for example, you need more time for better clearance from blood, it's insufficient. But with gallium-66, it's possible to make imaging not only at day of injection, but also next day. So it opens opportunity, additional. What is important to know about gallium? The first, we have a number pretty stable chelators which form complex, and minus log K means that it's a reverse logarithm of dissociation constant. In principle, it's 10 to 21 more likely to have the nuclide associated with those chelates than to be dissociated. The major issue is that blood protein called transferrin has dissociation constant, or log P, approximately in the same range. So it's very stable and very potent natural chelator for gallium, though its function is transfer of iron. And we can use it, and we will talk pretty soon about this. But essentially, what is important that it would constantly challenge our chelate containing gallium. From this point of view, the acyclic chelators are not reliable because they establish equilibria with surrounding liquid very quickly. So However, if you have macrocyclic chelator, they are thermodynamically approximately in the same range of stability. They are very inert kinetically. So the dissociation of gallium nuclide takes place very slowly, and transferrin doesn't have enough time to challenge that. Gallium forms stable complex with a number of other anions. And this is also an important feature. Problem is that majority of transitional metals, including gallium, lutetium, indium, all are relatively easily hydrolyzed in aqueous solution. So if pH is above 5, they start to form hydroxides, and hydroxides are not water soluble. When we're working with no carrier added radionuclide, the concentration of this hydroxide is not enough to form good precipitate. They would rather form colloids. But it's also bad because colloids are taken up by so-called macrophages, the residing Kupffer cells, residing macrophages in reticuloendothelial system in liver and spleen. So if we have colloid formation, we have very high uptake in the liver and the spleen, and that is a trouble. So we need to have the system which permits to keep metal ion in solution at least before labeling. And we typically use acetate complex for this purpose, because those complexes with acetate are strong enough to prevent hydrolysis and formation of hydroxide, but they are weak enough to permit transchelation to more potent EDTA or DOTA or NOTA chelators. However, the first clinical application has been associated with not a macrocyclic chelator, or even not a cyclic, it's a citrate, which is 3-dentate ligand, which occupies 3 positions of gallium coordination sphere. And the function of gallium citrate was essentially to keep gallium in ionic, not a hydroxide form, before it is injected in vivo. So what happened in vivo? Gallium very rapidly is transchelated from citrate to transferrin. And function of transferrin normally is transfer of iron, easy to deduce from its name. And iron is essential element. Particularly, iron is work as a coenzyme in synthesis of in the DNA elongation. Sorry. So that's important because site with rapidly dividing cells, that might be epithelium of intestine, but also tumors, produce a lot of DNA. So they need a lot of iron, and accordingly they overexpress transferrin receptor, which they use to catch transferrin. So if we manage to label a transferrin, then we can notice the place with overexpression of transferrin receptor and its tumors. So it has been approved once upon a time for detection of lymphomas, lung carcinomas, and melanomas. So one could use gallium-68, actually gallium-67, gallium-66 as well for imaging and detection of tumor stage. Problem is that it's also taken up actively in inflammatory diseases, which might be used, of course, for diagnostics of occult inflammation, but then it means that you could not distinguish between tumor and inflammatory sites. So the specificity is far not perfect. In addition, it is taken up in a number of sites that includes metal storage sites, ferritin in liver, and in some metalloenzyme. So specificity is compromised, but in early days of nuclear medicine, gallium citrate was very broadly used. It's still possible to purchase gallium citrate from some European suppliers, but it starts to be more and more exotic, and standard of care in majority of European countries is fluorodeoxyglucose, FDG PET. This is an example, and here is image of lymphoma, and one could see soft tissue metastasis in leg and lymph node, while you don't see, of course, in normal scan of it. But because there is no difference between inflammatory sites and tumor, so sensitivity is somewhat compromised. Gallium-68 is a short-lived positron emitter, and its function is for labeling and PET scan of somatostatin analogs and search and staging of neuroendocrine tumors. So historically, first label was indium-111, but nowadays indium-111 octoscan is essentially out of market. So neuroendocrine tumors such as insulinoma, carcinoids, mid-gut carcinomas typically overexpress a number of somatostatin receptors. And
Владимир, прошу прощения, можно вас перебить? У нас, видимо, проблема со слайдами, и многие участники видят только первый слайд.
Ой.
Мы не видим презентацию. У всех практически, да, я так понимаю?
Well, that's a problem.
Я уже перезагружалась, выходила, заходила, все так же, первый слайд.
У меня даже та же история. Первый слайд только.
Ага, okay. I will try to restart sharing. Do you see
No,
now?
no, I don't.
Do you see the slide?
Пока нет.
No.
Ой-ой-ой. Хорошо. It might be problem with the slideshow. I don't know. If we do like that, do you see slide now?
No, black screen.
Нет. Пока у меня черный экран.
Да.
Huh. Is it still the same?
Теперь
Now we
вижу.
see.
Sorry, do you see what is on the screen?
Да,
Да,
Yes.
теперь вижу.
да,
Okay, so
да, change.
it probably doesn't. Well, my experience with Zoom teaching is really limited. So, okay. So, gallium citrate. Here is the images of lymphoma metastasis, and which are not seen in normal scan. So, somatostatin analogs. So, somatostatin is important peptide in treatment of neuroendocrine tumors, and it's per se somatostatin is tetradecapeptide, which is involved in multiple functions, including regulation of gastrointestinal activity and regulation of production of gut hormones. It was found that short analog of somatostatin, octreotide or sandostatin, is as functional as sandostatin, and what is important, that somatostatin receptor type 2, type 5 are overexpressed in a number of neuroendocrine tumors. So actually, sandostatin or octreotide is used for treatment of neuroendocrine tumor. It's stabilized somehow tumor growth, but even more important, neuroendocrine tumor often have exceeding hormone production, and patients are suffering from many undesirable features like blood pressure drop, diarrhea, insufficient or exceeding insulin production, and so on. So, but also it is important to be able to visualize the site of elevated expression, because it's most likely would be a tumor, and in this way you could observe the extent of tumor metastasis and adjust therapy. Interestingly, that the first nuclide of gallium, which has been used for imaging of a labeling of somatostatin analogs, was gallium-66, more long-lived variant. And in this case, it was used for labeling of octreoscan and for labeling DOTA has been attached. DOTA is very versatile chelator, not ideal for gallium, but good enough. And this is very beautiful result of design. So normally, somatostatin is very vulnerable for blood peptidases. So it was re-engineered very carefully. So there are several types of peptidases. There are so-called hexapeptidases, which just cleave off amino acids from the ends, or there are so-called endopeptidases, which cleave peptide bond between some amino acids. So this radiopharmaceutical has been designed in that way that threamine acid has been at C-terminus converted to alcohol, so it was not recognizable by peptidases. At N-terminus, they place a DOTA, which is not amino acid, of course, so enzyme do not recognize it. And it was figured out that if you use D-tryptophan instead of L in this position, the somatostatin analogs retain the capacity to bind to somatostatin receptor, but is not recognized by peptidases. So it improved very much blood half-life of this radiopharmaceutical. The labeling conditions were very, very typical for labeling with radiometals. So it was used acetate buffer, and acetate buffer is necessary to prevent hydrolysis, very common buffer. pH might be lower, probably authors were afraid of going down in pH at that time. The short peptides tolerate very, very well high temperature. So 100 degree is no wonder. So it was 100 degrees centigrade, 15 minutes labeling temperature, and it provides the yield in the range of 85 to 95%. Well, in principle, it's possible always with radiometal to optimize the labeling condition to have over 95%, and typically further purification is not required. So current European pharmacia for somatostatin analogs requires radiochemical purity over 92%. So, but in principle, you can always use solid phase extraction, reverse phase, to purify free radiometal from peptide. So it was good enough after purification, and the authors here demonstrated that you can use gallium citrate and even visualize the tumor, but contrast would be quite poor. But if you use peptide, you essentially could see only tumor and no background from rest of the mouth. So the higher is the tumor to background ratio, the better is the contrast, the easier to detect a tumor. So it apparently has an advantage in imaging properties. But of course, the most important development was so-called DOTA-TOC, or DOTA octreotide labeled with gallium-68. The half-life of this nuclide, 67 minutes, is very well compatible with the rapid kinetics of somatostatin analog in blood and rapid accumulation in tumor. So this radiopharmaceuticals is now common tracer in neuroendocrine tumors, and one could see very high contrast imaging of liver metastasis of neuroendocrine tumor here. I hope you see slide. And of course, nowadays we always have PET-CT, which means that you could get an anatomical context of radioactivity accumulation, so you could easily localize where the tumor is, in which area. There were further development. I mentioned that DOTA is not an ideal chelator. It has minus log D, minus 20 D. You remember that transferrin is minus 20. Three other chelator called NOTA has much higher thermodynamic stability. Minus log dissociation is 30, so 10 orders of magnitude more stable than DOTA. And well, it's also macrocyclic chelator, so this means that it's also kinetically inert. So its best variant. The issue is in first generation was that in NOTA one of carboxylic group was used for conjugation to peptide, which was well, stealing one ligand from complex formation. But later on, another derivative of NOTA was developed, which is called NODAGA. And in this case, there is one carboxy group, two carboxy groups in one arm. So one carboxy group can be used for formation of amide bond with N-terminus of peptide, and another could participate in complexation of gallium. It's better radiopharmaceutical, I would say, though it's still DOTA-TOC is a workhorse. And the reason is that nowadays, nearly everywhere, somatostatin analogs are used in theranostic setting. So the clinicians first label DOTA-TOC with gallium to visualize tumor, and then label with lutetium beta emitter for therapy. And DOTA works both ways, both with gallium as with lutetium. NOTA or NADAGA, they have two small central cavity, so they work excellently for gallium, but not with lutetium. So the use of NOTA derivatives is suboptimal for clinical imaging. Another nuclide which played an outstanding role in development of targeting approach in medicine is indium-111. Actually, this is the first nuclide which has been used in connection with radiometal labels. The reason is historical. Among a variety of metals, indium has the highest stability in complex with DTPA. And DTPA was the first chelator which successfully been applied for labeling of antibodies. So that was the best nuclide at this time in terms of label stability plus nearly 3 days half-life of indium-111 is well compatible with relatively slow kinetics of monoclonal antibodies. So we know now that optimal time for imaging with radio-labeled monoclonal antibody is between 3 and 5 days after injection. Apparently, you need to have a long-lived radionuclide for visualization. But indium has a number of other nuclides which are potentially interesting. Indium-113M has a half-life of 100 minutes. What is interesting that it's also generator nuclide which can be produced from very long-lived tin-113. So some indium label colloids has been used in 60s and early 70s for imaging, and that's why the chemistry of indium has been studied quite well, and that is resulted in development of indium-111. So this nuclide played an important role in the past. Gamma energy is too high a little bit, so it's not of interest anymore. There is another indium radionuclide which is of interest. It's indium-110M. And it is positron emitter with a half-life approximately the same as gallium-68, which make it suitable for use as a positron emitter. There are some side gammas, no 100% positron yield, but in principle it can be used for labeling of somatostatin. We actually produce indium and publish a paper in Journal of Nuclear Medicine 2003, I think, where we have clearly shown that PET is better than SPEC. The problem is that you need to produce indium-110 by cyclotron, while gallium is available through the generator. So in the end, we of course lose a competition to gallium-68. There is yet another very interesting nuclide. It's indium-114M. So it's not a stable nuclide, but it is unique in that sense that it has very, very long half-life, nearly 50 days. It's very unusual for metastable nuclide. So it decays by isomeric transition, producing a cascade of low energy conversion electrons. And then its daughter is a beta, high energy beta emitter, which can be used for treatment of tumors. I mentioned once upon a time that different tumors have require different energy of particles for treatment. Small tumors should be treated with low energy gamma. Bulky tumor, non-operable tumor require high energy beta. The problem is that in the patient you might have, and you generally have, manifested both small and bulky tumor. So in this pair, mother nuclide emits quite a lot of conversion electron with energy 200, nearly 200 kilo electron volt. And that means that they act as beta particles. And then daughter emits high energy beta, and they 72 seconds, it's a very short time. So it typically would be localized in the same place where the mother. So there is opportunity for radionuclide therapy. But still, the most important nuclide, of course, is indium-111. And complexes of indium with chelator pretty stable. So I mentioned complex with DTPA minus log K is nearly 30. Stability of complex of indium with transferrin is much lower than in case of gallium. So it's a less challenge. And still macrocyclic chelators such as DOTA are preferable because of stability. Citrate and acetate forms less stable complexes, and acetate buffer is a preferable buffer for labeling with indium-111. Function exactly the same. Prevent formation of hydroxide and keep indium in a reactive form and suitable for labeling. So strong enough to prevent hydrolysis, weak enough to enable transchylation to DTPA or DOT, this nuclide. Oxine form quite stable complex, and this enables to use derivative of oxide complexes of indium for diagnosis of inflammation. So overall, indium is three-valent metal. Of course, either is hydroxide in normal physiological conditions, but even if you keep it with acetate, it's still charged complex, which is no good. So idea was to label white blood cells of patients, so-called autologous lymphocyte, with indium. So what happens? One takes blood from patients and isolate by centrifugation white blood cells, and then expose those white blood cells to complex of indium with oxide. So indium in this complex is has coordination number of 6, and oxide has 2 donating electron donating group. Is nitrogen having unshared electron pair, and here is phenolic group, which is relatively easily dissociated in phenols. So overall, it carries negative charge, single negative charge, and 3 oxide completely neutralize positive charge of indium and keep it in a stable complex. So you exposing indium to oxide, we convert charged and hydrophilic ion into neutral particle, which is coated with lipophilic aromatic moieties. So indium suddenly starts to be lipophilic. And that means that it could quite easily just diffuse through membranes of white blood cells. So because in form of oxide. Inside the cells, oxide react with cytoplasmic protein, which also quite often have chelating properties. Apparently, what happens then, the indium is transchelated to cytoplasmic proteins of white blood cells, and then oxide is just diffuses away from the cell, and indium stays inside the cell. So cells are washed then and re-injected to the same patient, and this is still the way to find occult inflammation. So there is quite extensive research.
See you tomorrow. Bye bye.
But still is ongoing research for good imaging probes for inflammation, but white blood cells remain by now, over more than 20 years of application, the workhorse in inflammation imaging. And of course, besides inflammation, you could see spleen here, because that's where white blood cells are ending up in edema, typically. I talked already about gallium DOTA-TOC, but in fact, the whole story started with this imaging agent called OctaScan. So it's the same somatostatin analogs. The difference between DOTA-TOC is that in this position there is phenylalanine instead of tyrosine. They are approximately equal in terms of affinity. And here is the DTPA, diethylene 3-amine pentacetic acid, where one carboxylic arm is used for conjugation to N-terminals. Generally speaking, DTPA is not really good chelator even for indium, because it loses indium to transferrin. So I would never label antibody using indium, but here is a special case. So blood kinetics of octreoscan is very rapid. So there is no time for transkylation. And suddenly the use of cyclic DTPA is good enough for labeling of octreoscan. On the other hand, advantage is that since it's acyclic chelator, you can label at room temperature, and keeping pH between 4 and 6, you get a very good yield at room temperature. So nowadays, this is supplied as a kit which contains lyophilized, freeze-dried octreoscan with chelator, the buffer components, and mannitol as a bulking agent. So it's so-called two-vial kit. When supplier sends you one vial with octreoscan and one vial with indium 111. In situ, in hospital pharmacy, you put them together, incubate for half an hour at room temperature, and then typically your radiopharmaceutical is ready. So you need to take some amount to test labeling, yield, and purity, and then you can use it for imaging. And these are historical images. This is one of the first images of neuroendocrine tumor, and this is representative image of patient with neuroendocrine tumor, and you could see multiple metastases in his body. Everyone, a black dot is a metastasis. Not as sharp, not as contrast as gallium PET, but good enough to be a workhorse of treatment and diagnosis of neuroendocrine tumors for more than 2 decades. So now it starts to be competed out by gallium 68 variant. There are several antibodies which have been approved by Food and Drug Administration for radionuclide imaging. For detection of tumor, it was indium labeled Oncosint, an antibody which detects so-called TAG72, tumor-associated pancarcinoma antigen. Prostacint detected PSMA, the same as now fashionable binders. And myosint is specific to myoglobin protein in myocytes in cardio cells, which use it as a reversible operative storage of oxygen. So in normal cells, myoglobin is of course not visible from outside, non-accessible from outside, but in the case of myocardium infarction, the cells die, and necrotic cells, myoglobin is exposed to blood flow. So an antibody localized in area of necrotic myocardium. Well, the problem is that they all have been approved by FDA just before approval of FDG, fluorodeoxyglucose, which provided generally better specificity and much better sensitivity in comparison with antibody. 5 years ago, the only indium-labeled antibody on market was prostacid, but I guess it's just gone because of new generation of binder of PSMA. What concerns therapy? I have mentioned already that you need to have a nuclide with different energy of beta particle to treat either small or bulky tumors, and probably with different half-lives. And for the moment, there are radiopharmaceuticals labeled with high energy beta, yttrium-90, low energy beta, lutetium-177, and intermediate beta particles emitter, samarium, on the market and approved by regulatory agencies. Zevalin is used for treatment of lymphoma. So lymphoma is B-cell malignancy when B cells are overproduced, so it starts to be malignant, and therefore one tries to treat lymphomas with radiation. So fortunately, lymphomas are generally very radiosensitive. To kill a lymphoma cell, you need to deliver 5-fold less activity in comparison with solid tumor. So yttrium is good enough. Antibody Zevalin is murine antibody, quite unusual nowadays, but there is a good rationale for using in this case. So typically, those patients are heavily pretreated and severely immunocompromised for this reason. So that means that formation anti-murine human antibody is not very likely. On the other hand, using murine antibody in human, you typically have a reasonably rapid blood clearance. Good engineered human antibody might have half-life in vivo of 19 days, which too much for therapeutic antibody. Murine is good enough. So they used the variant of benzyl DTPA. It's just extra methyl group, which a bit restricts freedom of rotation of DTPA and stabilize complex appreciably. And it has been approved by Food and Drug Administration for human use in September 2009. I would say that history of Zeppelin was probably first bell of unhealthy situation in healthcare in capitalistic world. We see now coronavirus situation, but Zeppelin demonstrated all disadvantages of situation where healthcare is a business. In fact, it was appreciably better than rituximab, cold variant, for treatment of lymphoma. The problem is that normally such patients are treated by oncologist or hematologist. In the case of cold antibody, he or she takes this antibody from the shelf and inject in patient and get all the benefits of treatment. If one uses the radio-labeled antibody, they need to remit to nuclear medicine and lose their income. You could guess what happens. So expectation was that annual sale of Zeppelin would be at least $1 billion. Turned out that it was 20 million, 50-fold lower. This antibody was marketed by Bayer Sharing. It's a bloody, economically strong, big pharma corporation with a lot of opportunities. They try to act through patient organization and so on, but against the greed of clinicians, they could not do nothing. So there were two antibodies for therapy, Bexar and Zeppelin, approved at the same time. They are severely underused. Bexar was withdrawn from market, Zeppelin survived, but barely. So it's not only matter of doing or producing right imaging agent, unfortunately. It's a problem of healthcare organization. So Zeppelin lost its fight. Phosphonate complex of samarium is used in bone pain palliation. So it utilizes feature of new form bone to accumulate phosphorus or phosphorus compound. So when there is a bone metastasis typically destroys the bone around. And bone tries to counteract by forming new bone tissue, which requires a lot of phosphate or phosphonate. So in this compound, samarium is complex by phosphonates, and phosphonates play dual role. First, they keep samarium 153 inside the complex, and second, they find the site of increased bone formation, which means around bone metastasis of cancer, and then it locates close to malignant cells, and medium energy beta of samarium are irradiating tumor cells and suppress their growth. One more important nuclide is lutetium. Lutetium 177, 6.9 days half-life, medium beta energy, and or rather low beta energy, and this is advantage. It turns out that kidneys tolerate much better nuclides which emit low beta energies and high beta energy. An explanation is simple. The most radiosensitive part of kidney are glomeruli, but nuclides typically localize in proximal tubuli. So if nuclide has a high energy beta, it's enough range in vivo to over irradiate the glomeruli, but for example, it's a case for yttrium. In case of lutetium, beta energy is not enough to irradiate glomeruli, so kidney experience much less toxicity at the same dose, absorbed dose, as with yttrium. And here is a magic example. It's given me by my friend Dr. Ulrika Garsky. So he labeled somatostatin analog, closely related to octreotide. It's a dotatate. So it's the same peptide, though at C-terminus there is acid, not alcohol. And DOTA permits labeling with lutetium. She told that this patient was scary awful. His liver was so full of metastasis. Whatever you see here, it's a liver metastasis, except from this hot spot. So liver of this patient, because of metastasis, filled the whole stomach. This hot spot around liver is a urinary bladder, which was forced by metastatic liver nearly to midgut. So, but cycle by cycle, you could see the accumulation of activity in liver metastasis decrease. So patient survived and liver shrink in size and got a nearly normal. So that is fantastic effect of therapy. This patient had no other treatment option. The metastases were too numerous and too big for any surgery. Now, it was downsized so much that the metastasis could be operated. I would say this is a spectacular example. To be honest, therapy, radionuclide therapy is still not a curative. It's mainly palliative, given to extend the survival and improve quality of life in patients. It has also to be noticed that neuroendocrine tumors are awful by their side effects. So production of hormone disbalance is something which compromises life of patient enormously. And even there is no big shrinkage of neuroendocrine tumor, but very often there is a so-called chemical response, so stop of production of hormones. The quality of life of patient improves enormously. So this is an area where we can make an impact. I know that Tomsk Polytechnic University establishing now production of lutetium for therapy, and that is really, really great. So we can improve life of patients. Importantly, of course, that this was story of success, which helped to keep development of radionuclide therapy, despite, for example, failure of Zeppelin. What is important is to remember that chemistry of radio labeling with metals could influence the biodistribution and targeting properties. For example, was shown that earlier that if you conjugate daughter to somatostatin analog, they will be internalized more rapidly than the same analog conjugated to DTPA. In this interesting study, they use tyrosine derivative of octreoscan, so they put a label, not a radiometal. They put a radioiodine label in tyrosine, and then try to see how the internalization take place. And that was first documented case that chelators influence cellular processing. Also, radiometal influence the biodistribution and targeting properties. We can come back a little bit later on that. So it was demonstrated in dual isotope studies that if you label daughter nucleotide with yttrium, it will be higher uptake in tumor in comparison with indium. And I will talk a little bit about influence of metal chemistry on targeting properties using my favorite affibody molecules. So affibody molecules is so-called engineered scaffold protein. It's non-immunoglobulin alternative to monoclonal antibody, for example. So they have been designed based on protein A scaffold. Protein A is part of anti-immune defense of staphylococci. So staphylococci would like to camouflage themselves, to be not recognized by host immune system. So what does they produce on the surface? Long chain of protein A will just catch the host antibodies by the FC domain and create a coat of host antibodies around themselves. So they camouflage in this way. So protein A is used a lot in biotechnology, for example, for purification of antibodies. In this case, a team of Professor Ulen in Royal Institute of Technology in Stockholm just took one domain, and what did they do? They randomized 13 amino acids which normally are involved in binding to Fc region of immunoglobulin. So it's three helix protein which folds spontaneously. It does not contain any disulfide bonds inside, so it just folds. And by randomization was created a library which now contains 20 billion members. So it's comparable with whole repertoire of human antibodies. So from this library, one can select binders which bind with high affinity to different tumor-associated and non-tumor-associated proteins. Antibody molecules are roughly 20-fold smaller than immunoglobulin. So normal IgG antibody has a molecular weight of 150 kilodalton. Antibody molecules is 6-7 kilodalton, so it's much smaller. And the small size enables to extravasate rapidly, go from blood flow into tumor to extracellular space. Also clears very rapidly from blood, but affinity is as good or might be better in comparison with antibodies. And you can produce them either by peptide synthesis or recombinantly. And well, we have shown that as one of advantages is that if you label full-length IgG 6 hours after injection, you could visualize the tumor. But generally, it's a lot of activity in blood pool. So this is a heart. These are carotids in mouse. And of course, liver, frequent metastatic site is also full of blood. So you need to wait several days to make a good visualization. With antibody 6 hours after injection, you have very high contrast imaging, which is expected. Here is one of our examples how we try to optimize the antibody molecules. This case is synthetic antibody molecules which contain at N-terminus simple or straight 3 different chelators. It was 3 other chelators nota, tetra other chelator dota, and 3 other chelator nadaga, which is similar to dota, to nota, but it has one extra carboxyl arm, which permit to have the same charge as dota. What we have found that the difference in charge play important role. So distribution in normal tissue was nearly equal, but positively charged dota had much higher uptake in liver, in spleen, and in intestines and in bones. So 58 amino acids plus chelator on it, but single charge at N-terminus and charge in the chelator play a role. And this has been confirmed in study in tumor-bearing mice. And we could see that even if you compare only those two chelators, you have tumor to blood ratio significantly higher in case of Nadaga compared to DOTA. It is factor 2. Believe me, this is a lot. So selection of right chelator can help you to optimize imaging properties. Also, you could label it's a question of composition of chelator, but you could place chelator in different places of targeting protein. For example, here we studied again synthetic antibody molecules where chelator was placed either at N-terminus or at C-terminus or in the helix 3, but not in the terminal position. Here as well, we have found quite big influence of label position. First and foremost, we figure out that placing in the helix 3 is no good. Most likely it destabilizes the whole helix. Affinity actually dropped quite appreciably, and tumor uptake accordingly was decreased. But we could see also significant difference depending where we place our chelator at N or NC terminus. And this was transformed in further increase of tumor to blood ratio, varying with the C-terminal placement provide initial gain of factor 2 compared to the N-terminus placement. So overall, positioning and composition of label play a role, and it can affect both the uptake in normal tissues, uptake in tumors, and in this way determine a contrast of imaging. This is quite known phenomenon, and also that I mentioned already the different chelators have different nuclides in the same chelator might have quite a noticeable difference in biodistribution. And the reason is in the difference of the coordination sphere. For example, do you hear me? All right. So the difference is that, for example, yttrium or indium, they have coordination number of 8. So in their complex with DOTA, they take all 4 nitrogen in equatorial plane. So normally they would have also all 4 carboxy oxygen. If one arm is used for conjugation to tumor targeting agent, carboxy oxygen is missing, but carbonyl is still there, and they used weak surrogate. But what is important that it's a very closed system in this case. In the case of gallium, story is different. Gallium is small and its coordination number is only 6. So with the same daughter, it has only 6 donor atoms, and these are the same for nitrogen from equatorial plate, but only 2 carboxylic arms. So one arm is used for conjugation, and one carboxylic arm is just stick out, protruding with negative charge. So accordingly, charge distribution inside this complex is very different compared to charge distribution inside the complex of yttrium or indium with the same daughter. And charge distribution can have a profound influence within the molecule, so preferable conformation, and so on. And it results in different properties, and it affects affinity, and it affects the off-target interaction. The first work has been done by a team of Professor Mecke in Basel, where they show that gallium and indium of somatostatin analogs influence quite a lot of affinity and distribution properties. Initially, I was skeptical that it should have as profound influence with affibody molecules, because affibody molecules roughly 6-fold bigger. It didn't look that very probable that just subtle change of complex geometry would have a pronounced effect. Still, since we were warned by work of Mecke, by Marion de Jong's work, so we, just to be scientifically thorough, investigated this effect with affibody molecules, and now it's very much clear that we have to evaluate all thinkable chelators and all thinkable nuclides for them in order to find the best one. Because the difference in contrast, depending on position and composition of chelator and matching radionuclides might be several-fold different. So, radiometal chemistry is very rich. We have a number of radiometals for imaging. We have a number of radiometals for therapy. And it's important to keep in mind that labeling chemistry is not only a way to stably attach a radionuclide to our tumor-seeking moiety. It's also potential to improve the biodistribution. So selection of right labeling chemistry for radiometal might make difference between success and failure. That's it for today. Спасибо за ваше внимание. Если есть какие-то вопросы, я готов ответить, и я готов это сделать по-русски.
Спасибо.
Есть ли какие-нибудь вопросы? Какой я молодец. Однако все понятно рассказал, или как?
Можно один вопрос?
Конечно.
К последнему слайду. Координационное число, получается, для металлов оно будет в данном случае постоянно или могут варьироваться? Просто обычно несколько приводится.
Координационное число может быть разным. У того же Индия это может быть 5, 6, 7 и 8. В принципе. Оно также зависит от свойств лиганда тоже. Вот. Но у Индии наиболее стабильные комплексы имеют координационное число 8. У Галлия это 6.
Это опять, наверное, все термодинамическими параметрами, да, определяется?
Ну, да, там можно, ну, термодинамика определяет, извините, вообще все. Хоть убейте. Вот. Не всегда ее легко посчитать. Вот. Ну, известно, что если металл является жесткой кислотой, то он предпочитает мягкие основания и наоборот. То есть это тоже, то есть это уже правило большого пальца, но в общем оно нормально в этом смысле работает тоже. Вот. Ну, из Индии любит азот и любит карбоксильные кислороды. Галлий вполне еще поверх этого работает с фосфатами и с фенолятными кислородами и так далее. То есть, ну, это очень большая литература, которую, в общем, впихнуть в одну лекцию дело немыслимо. То есть я просто постарался здесь сделать некий обзор тех нуклидов, которые наиболее часто используются в ядерной медицине.
Спасибо.
Это было легко. Еще вопросы есть?
Там в чате есть вопрос.
Я не очень хорошо слышу.
В
чате вопрос.
В чате. У меня одна беда, что я, когда имею share, я не вижу чат. Вот какая штука. I had a question from Nelson concerning the use of is indium currently being used more than technetium-99? Answer is no. Technetium-99 is for the moment the most frequently used radionuclide. And reason is that one could, of course, say that it has gamma energy the most suitable for gamma camera, but it's vice versa. In fact, currently gamma cameras and SPECT cameras are optimized to make best image with technetium. And there is a second major advantage of technetium. It's cheap like dirt. Once upon a time, when we had to make a decision how to label F-body molecules, I asked prices for radionuclide, for technetium, and for indium. So standard price for single dose of indium was 4,000 crowns, Swedish crowns. Well, it was Swedish hospital. So, and this was one patient dose. At that time, if you pay twice more, you can get 32 gigabecquerel of technetium generator, and you could run hundreds of patients. So let's say price is at least 50-fold different. So for technetium it's lower. And it's a very powerful driver to develop chemistry or to develop technetium radiopharmaceutical. Disadvantage, I will talk next time. The radiochemistry of technetium is crazy. It's 8 different oxidation states, and the final oxidation state depends on reducing condition, it depends on chelator, it depends on chemical surrounding. It's not easy to work with technetium. But but on the other hand, it's very cheap nuclide, and it's preferable nuclide wherever. But still there is a space for other nuclide, not only for technetium, but for other. But if you look now what happens in Tomsk, essentially the camera is optimized, SPECT camera is optimized for technetium, and next generator is optimized for technetium. And nowadays you get standard collimator for technetium. For indium, if you want to make indium labeling, you need to order a special collimator for that, for extra money. Earlier it was included in price, now not anymore. But there are some things which are easier to do with indium than with technetium. Other questions? Well, I hope that everything is clear. I really apologize for the mess with presentation. I don't know what has happened, and well, my experience with Zoom is of course very limited, so but I'm happy that we managed to fix the problem. Спасибо большое за внимание. Я
Спасибо вам.
Ну ладно, а на следующей лекции мы поговорим как раз о химии технеции. Она интересная, она не простенькая совсем. Но в общем, даже если хоть чуть-чуть научишься с ней работать, там можно делать очень интересные вещи. Я его и люблю, и ненавижу одновременно. Ну, хороший. Ладно, до свидания всем.
Спасибо. До свидания. Спасибо.
До свидания.
До свидания.
До свидания.